Hello and welcome to the mind map about viral disease. Okay, we're going to zoom on in. Okay, first we're going to talk about the characteristics of viruses. They are acellular because they're not made of cells. They're very, very small because they're only about 5% the size of a bacterium. They have a protein coat called a capsid, and some can have an outer lipid envelope. They all contain genetic material, which is either DNA or RNA, but they cannot self-replicate. Once inside the cell, it uses the cell machinery to replicate, and a baby virus is called a virion. All viruses possess proteins that I call receptors that allow it to attach to host cells. These receptors determine host specificity, which is the host that it will invade, and there is wide diversity in the mode of transmission. So if we look, we've got a standard virus on top, and we've got one that has a lipid envelope below. Here is a lovely picture of the virus life cycle. Number one, we've got attachment or docking. This is when the proteins on the virus attach to the proteins on the cell. Upon attaching, it then internalizes in a process also called penetration. Then it undergoes a process called uncoating, where the capsid is removed to release the viral genetic material. In this case, it says RNA. Sometimes the RNA can undergo reverse transcription to be put into the DNA, or sometimes the viral RNA can be used inside the cytoplasm. Either way, it's used for transcription and translation. After translation, the proteins are reassembled in a process called assemblage, and then it's released. And in this case, part of the cell membrane is used to create the lipid envelope. The release of the virions also causes the death of the cell, and the death of the cell is the thing that causes the symptoms of the viral diseases. I thought I would stop here and you guys can get an idea of the relative sizes of things. So first we've got a red blood cell, which is quite large compared to a virus. About a quarter the size of a red blood cell is a bacterium. But then at about 100 nanometers, we've got viruses like influenza, HIV, and COVID. We've also got the 7 steps of the viral life cycle, which is docking, entry, uncoating, replication, protein synthesis, assemblage, and release. Okay, next I've stopped at this particular place where we're going to look at pathogenicity. Pathogenicity refers to the ability of the pathogen to cause disease. Viruses only infect certain types of cells. We call this the tropism. For example, influenza only affects respiratory cells. Viruses only affect the cells that have a complementary receptor. For instance, influenza A only infects human respiratory cells. As mentioned earlier, viruses cause damage by cytolysis. This is when the cell ruptures to release the baby virions, and therefore the cell no longer has a function. Some viruses hide from the immune system inside the cells. For example, HIV hides inside T helper cells, otherwise known as CD4 plus cells. A very similar term to pathogenicity is virulence, except virulence is the extreme of pathogenicity. Whilst pathogenicity is measured in the amount of damage, virulence is measured in the amount of deaths caused by the pathogen. Okay, the first virus we're going to look at is called the Ross River virus. It's an arbovirus. It's transmitted by arthropods, and in this case, mosquitoes. It's a single-stranded RNA virus. It has a lipid envelope taken from the last cell that it was in. The reservoirs are marsupials, primarily wallabies, and birds. The mode of transmission is a mosquito bite. The incubation period is between 3 to 11 days. Now, with the symptoms, they're quite general, but the one that stands out is polyarthritis. Polyarthritis is the inflammation of many joints all at once. The duration can be weeks normally, but in some cases it can last years. Treatment is rest and anti-inflammatory drugs. Prevention is the control of mosquito numbers and personal behavior like wearing loose clothing and not going out at dusk. Okay, next we're going to talk about influenza. Again, it's a single-stranded RNA virus with a lipid envelope. It has 2 surface proteins of note. It has hemagglutinin, which is HA, which is an attachment protein, and when HA binds to the surface proteins, it allows for internalization. The other one is called neuraminidase, or NA. It's a protein that allows for the release of the virus. It allows the virions to take a bit of the host cell membrane to make its lipid envelope. Influenza's tropism is respiratory epithelial cells, and it has an affinity for humans, pigs, birds, and horses. It spreads rapidly through respiratory droplets of all sizes. It's highly infectious, so that means it's easily spread, but with low virulence, which means it doesn't kill many of the hosts that it lives in. It undergoes antigenic drift, which are small mutations in the surface proteins to create the different strains like H1N5, etc. Occasionally, there's an antigenic shift, which allows for influenza to spill over into other species, which means it jumps from one species to another because the receptors have mutated enough to do so. Influenza has an incubation period of between 1 to 4 days. When you get it, it normally lasts about 2 weeks until full recovery. Symptoms include fever, a productive cough, malaise, which means you don't feel like doing much, and having body aches and pains. Treatment is antivirals to prevent protein synthesis. Prevention is vaccination, quarantine, and isolation. Now, honeybee viruses aren't one particular virus. There are several that we must know. There is deformed wing virus, which is a single-stranded RNA virus. It uses the vector Varroa destructor, which is a mite. It lives in the nervous system of the honeybee. Transmission is through direct contact. It's vertical transmission, which is the birth of the larva and then maturation into the pupa. Both the larva and the pupa need to be fed, and this is when transmission occurs. Symptoms include vestigial and crumpled wings. Vestigial means they have no use. The bee is paralyzed and death rapidly follows, and this can cause colony collapse. Acute bee paralysis virus is also a single-stranded RNA virus. It can use the vector Varroa destructor, but not always. It lives within the nervous system of the honeybee. Again, transmission is through direct contact while feeding the young, but in particular the virus lives in the salivary gland secretions, which are then passed on between the different bees. Symptoms include ataxia, which means the bee can't stand up, trembling, it can't fly, it's bloated, and it normally dies within a day because it's acute. But then we've got chronic bee paralysis virus, which is very similar to the acute virus. It's a single-stranded RNA virus, but it doesn't involve the vector Varroa destructor. It lives within the nervous, digestive, and reproductive systems of the bee. Transmission occurs when there is direct contact with dead bees. The symptoms include ataxia, trembling, not being able to fly, having a bloated abdomen, but this time death occurs within a few days. The last two honeybee viruses are black queen cell virus and sac brood virus. The black queen cell virus is again a single-stranded RNA virus. It doesn't involve the vector Varroa destructor. It lives within the digestive and reproductive systems of the queen. Transmission occurs during birth, so once again, it's vertical transmission. Symptoms include the black cell walls of the sealed queen cells. The pupa turn yellow, then black, then death. With sac brood virus, again, it's a single-stranded RNA virus and it doesn't involve Varroa destructor. It lives within the digestive system of the honeybees. Transmission involves the nurse bees that contract sac brood virus when removing the dead pupa. And then it passes on to the next pupa whilst feeding them. Transmission also involves the activity of foraging bees that can pass on the virus to other bees with contaminated food during communal feeding, which means that one bee will feed another bee. It's a very social activity. Symptoms include the pupa turning yellow after capping, then there is a failure to mature, and then death follows. Okay, I've stopped here so that you can see what a Varroa destructor looks like. This is the mite on top of a pupa, so it's very, very small, but very, very destructive. It actually bites into the pupa or the adult bees and drinks the hemolymph, which is the fluid inside the bee that acts like the blood of the bee, but it's not contained within blood vessels. And then below that, we've got the life cycle of the bee. The queen lays the egg. After 6 days, it turns into a larva. Once the larva reaches full growth, capping occurs. The larva then turns into the pupa, and after 21 days, an adult bee emerges. Thank you for listening. I hope this was helpful to you. Don't forget to like and subscribe, and I'll catch you later. Goodbye.